I would like to thank the organizers for putting together an excellent meeting, and thank you also for having me. I'm Babak Momeni. I'm from the biology department in Boston College. Before I start, I would like to acknowledge people who actually did this work. Helen, a former postdoc, Sam, a former graduate student, and 3 talented undergraduate students who did the majority of the analysis. And also the funders, the Smith Family Foundation and NSF. In our lab, we're interested in studying microbial colonization resistance and engraftment. And our main motivation is from the perspective of the nasal microbiota. I won't talk about our work, experimental work on nasal microbiota today. I would love to discuss it with you. So if you're interested, let me know. I'm happy to talk about it. But in that context, there are 2 important applications. One is protection against pathogens. So microbes that are in our nose are thought to be protecting us from pathogens that we are constantly getting exposed to. There are still around 13 billion respiratory infections happening per year, so there's room for improvement. But they do their job and they don't get credit for infections that don't happen. The other side of it is when the situations that we want to intentionally introduce a strain into that environment. So let's say a probiotic, maybe for drug or vaccine delivery. And this is the opposite side, that there is a community that naturally is resistant to invaders, and we want to intentionally introduce something new into that environment. And right now, we don't know how to do that properly. From a theoretical perspective today, I'm going to ask this simple question. How does the spatial structure affect the species coexistence and colonization resistance in a microbial community? We will limit ourselves to species interactions. The species interactions is known to influence coexistence and colonization resistance. And there are other factors. For now, we will focus on this particular aspect. And I'll give you the upshot that we find that modulation of interaction strength and changing the diversity of available niches is what ends up to matter in these communities, and space contributes that way. So for the rest of the talk, I'm just going to provide more evidence for these 2 factors. The sort of model system that we use here is a computational one, and it's structured fairly sort of intuitively. There are species that interact through mediators that they are producing into the environment, and these mediators can positively or negatively affect other species that share the same environment. So there are some species present, they produce some compounds into the environment, and those compounds affect other species that share the same environment. And in this particular case, because we are interested in spatial structure, we put them in a 1D spatial matrix. Essentially, each species will have a distribution in that 1-dimensional space, and they produce metabolites that will have also a spatial distribution. To decide how to structure our model, we did very simple experiments by putting microbes under the influence of different positive or negative factors. So for example, at different concentrations of antibiotics, we measured their growth rate, or we added something that would improve their growth rate and measured how the growth rate was modulated. And I'm showing only 2 snapshots, but we see these patterns over and over again, that growth rate linearly drops when we add more inhibitors into the environment, and it grows in a saturable form when we add things that promote their growth. And we incorporate these 2 assumptions into our model. And the overall model looks like this. It looks a little intimidating, but there isn't really much going on. There is, let's start from the top one, the distribution of species. We assume that the species disperse into the environment passively, so we still assume that process follows diffusion. And there is some local capacity on how much that local space can accommodate more cells, and they grow as they get inhibited or facilitated by the compounds that are present locally at that particular location of space. And the mediators, again, diffuse into the environment, follow standard diffusion, and they get produced and consumed by some species and consumed by other species. To assess coexistence, what we do is that we put an initial pool of microbes that are randomly interacting together. We go through rounds of growth and dilution until this community reaches a stable state. And we look at the richness of those communities, and by repeating this process over many random interaction networks, we take the average of richness of those communities, and mean richness is, for us, a quantifier of coexistence. Some communities reach higher richness, some lower, but on average, we say that mean richness would be a descriptor. And here is a simple comparison, and just to orient you, the square ones, which might not be easily seen, are from the spatial communities, and the filled circular ones are from the well-mixed community. And in these cases, the interaction network is intentionally exactly the same. The only thing different is that in one case, they are simulated as if they are well-mixed, and in the other case, they are in this one-dimensional matrix. And you can see here that spatial communities in this case show higher coexistence. The other thing to notice is that along this scale, we are changing the proportion of facilitation to inhibition in the initial pool of microbes that we were putting together. And you can see that the effective space becomes more pronounced when there are more facilitation interactions within the community. We then started asking how this coexistence in a spatial community would depend on the parameters of the system. And the first one that we looked at was the dispersal rate of a species in that environment. Yes?
The question, richness, is it just a number of species? So it is between 2 and 3. It's relatively low rich. Well, I mean,
right.
What is the definition of richness?
Right. The richness is the number. In this case, I think we start from 15 species that we put together, and we get some communities that are 7, 8, maybe species, a lot of communities that are 1, 2, 3.
So on average, it's between 2 and 3.
Right. Right. So that's what we get in this case. If you increase the number of species in the initial pool, this number goes up, but not by much. It's typically very hard from this setup to get communities that are very high richness. So the first thing that we see is that at low dispersal rates, we get spatial communities that are that have higher coexistence compared to well-mixed communities. And for these ones, I'm showing all different cases, but maybe for simplicity, you can focus on the 9E210 that has more facilitation in there because the effect is more pronounced in that situation. When we go to the other extreme and make the dispersal coefficient infinite, which is essentially distributing all the species immediately all across space, we see that spatial and well-mixed become the same as you would expect. At intermediate levels of dispersal, something funny happens. We didn't expect it, but this is what we see, that the coexistence of spatial communities goes down. I'll take a slide, but I'll come back to it. The first thing that I wanted to emphasize was that just asking how this dispersal is affecting the coexistence. And we think that there are two different factors acting. One is that, let's say this axis is the as we go towards lower dispersal or more spatial localization. We get more pools of more local pools when we do that, because species are not dispersing much. They end up interacting just closer to interfaces with other species. So you have a lot of these local pools of species interacting with each other. In the other case, when you have faster dispersal, these species are spreading in space. So you end up with pools that have more species in them, but you have fewer of those pools. So there is this trade-off that between having pools of larger sizes versus having more pools of smaller sizes. And it seems like in the end, the effect of having more local pools is more dominant. So we get more coexistence in these communities. So let's go back to intermediate levels. We looked at different factors that could potentially explain this, and we ended up seeing that this one was the one that made a difference, that there are self-facilitation interactions in these communities. So production of a compound that benefits the same producer. And these self-facilitations are kind of strange. It's a sort of a positive feedback for that organism. And if in some cases that can lead to dominance of those organisms, which means getting rid of every other organism in the community and lower coexistence. So what we did to test whether this is an important factor or not is that we went back to these communities, took those self-facilitation links, and just made those links weaker. And then we recalculated all the coexistence factors with the original cases and these weakened self-facilitation cases. What we see is that at low dispersal and well-mixed cases, nothing changes. But at intermediate levels, we see that makes a difference. And our interpretation is that at low dispersal, this effect can be wrapped into just the fitness of that organism in that environment. It's not interacting with anything else. If it helps itself, it's as if it has just higher growth rate. And in the other limit, then that everything is well mixed, the return of that benefit to the self is less because nutrients are distributing into the environment, so less of it can affect the same species. But at intermediate levels, these cells are creeping into the niche of other species, and because they're still receiving the self-benefit, they dominate those environments and run other species extinct. So that's what we think is happening in this case. We then moved on to looking at the diffusion of mediators. So mediators in this environment are also diffusing, and we asked what happens if we modulate that factor. And again, maybe focusing on the black and gray ones in this case for the cases with more facilitation, you can see that as we increase the diffusion coefficient for metabolites or mediators, we get higher coexistence. And this was also a little bit strange to us. Sort of in my head, when we started this project, I had it fixed based on the literature that I had seen that the more localization you have, the higher coexistence you get. And in this case, that doesn't seem to be the case. And what we think is happening is that as you make these diffusion of mediators faster, you are again running into this trade-off that in one case, when the mediators are slow, you have more localized interactions. And in the other, but I guess the flip side is that when mediators are diffusing faster, the strength of interaction with species that are a little bit further apart becomes stronger. And that increase in interaction strength dominates in this case and leads to more coexistence when you have faster diffusion of mediators in the environment. Does everything make sense so far? Oh, we lost anybody. So we then moved on to colonization resistance. So we have, I'm not sharing everything. So there are more things that we looked into for coexistence in the spatial structured environments. But we also wanted to know how being in a spatially structured environment affects colonization resistance of species. And the assay that we use is a relatively simple one, but it's also based on experiments that we run in the lab. That again, we start from an initial pool of microbes. We propagate them until they reach some stable state. We call that the resident community. We introduce the invader, continue that for about 100 generations, and ask what happens to the community. And there are two different axes. One is whether the invader is successful or not, and the other one is whether the original members of the resident community are still present or not. So you have, on top, you have either the maintenance of the resident richness, or it drops because some of the existing members go extinct. And in the other dimension, you have either maintenance of invader or it goes extinct. And you have four outcomes, either augmentation, that something is added to the community, or displacement, that the invader is added, but something that was already there is gone away. So invader replaced somebody. resistance when the invader goes away and the community stays intact, or disruption when the invader cannot establish, but in the process of invasion knocks out some of the members of the community. And we typically look at these 4 outcomes in our simulations to see what happens. This is a little busy, but bear with me, we'll go through it together. So in this case, what we are doing is we are looking at the difference between well-mixed and spatial communities with the additional axis here, which is the ratio of consumption and production rates. So this is another factor that we have looked into. I just wanted to show you some of the results for that too. And the thought is that if you increase the consumption to production ratio, then the interactions become more localized. Things are quickly taking up the nutrients that are getting released. So there's a short range of distribution of those mediators in the environment, and everything will be a little bit more localized. And you can see that typically in these cases, we see that augmentation increases in a spatially structured environment, displacement increases compared to well-mixed, and resistance will be lower. So these communities will be less resistant to invaders when they are in a spatially structured environment. And that effect will be more pronounced when consumption to production ratio is high, which means interactions are local, and there are many available biotic niches in that environment. The other factor is we also looked at the, again, the diffusion of mediators. And if you, again, the x-axis is the same, sort of looking at the localization, but the colors represent different diffusion coefficients for the mediators. And sorry about the colors, but overall the trend is that as you increase the, let's say here, maybe it's the easiest one to see, as you increase the diffusion rate for the mediators, resistance will increase. Did I say it right? Or maybe rephrasing it is the weaker interactions happen at the slower mediator diffusions, and that strengthens colonization resistance. You might have already noticed that I'm kind of saying the same things that I had said before, but in a different context. That, for example, here, what we expect is that when we have faster diffusion of mediators, then it increases coexistence, and increased coexistence kind of intuitively makes sense. That makes it easier for invaders to invade, or weakens colonization resistance. And those are the patterns that we've seen. So instead of going through other parameters and showing you the same thing, I instead offer this alternative that we think coexistence, or reinterpreting, is survival fraction from a pool of initial microbes is tightly linked to invasion success. And this has been seen in this paper from the Gore lab that they ran Lotkov-Volterra type models, surveyed a range of parameters, and they saw that there is this correlation between invasion probability or invasion success in our language and survival fraction or coexistence in our language. We did the same thing in consumer resource model, and we see the same trend. And in their paper, they also have experimental data that supports the same trend. So I guess what I would like you, I guess, to accept at this point is that this is probably something general that you might see in other systems as well.
But I thought if you have higher coexistence, you might be able to prevent further inoculation. So that is sort of like counter to.
So I think there is some maybe inaccuracy in my language. Higher coexistence in our case means what fraction of the initial pool of microbes we can can survive together. So communities that already exist in the environment, that's a slightly different story, and I'm happy to talk about it with you later. But I think that should be treated differently from these incipient communities that are emerging from enrichment process. I think I'm on time. So two simple takeaways for you. One is that in spatial communities, the strength of interaction and diversity of available niches are the two factors that you should care about. These are the things that I think underlying the properties that we see in these communities. And I would suggest that to gain better intuition about coexistence, we should try to take whatever parameter of interest or property of interest that we are examining and map it into these basic already known ecological principles that describe coexistence. And I think that's a helpful framework to interpret some of the data that we see and also to be able to make predictions about other cases. And the other takeaway is that factors that promote more coexistence, it seems that they are also contributing to higher invasion success and less colonization resistance. So that makes things a little bit easier. We don't have to think about two completely separate ideas and concepts. We can just compile everything together and think of them within the same framework. Thank you, and I'm happy to answer any questions you might have.
Do we have any questions from trainers?
Thank you for your talk. You mentioned at one point that you assume that locality would increase coexistence, and then your explanation was that in the non-local limit, the positive mediators just diffuse everywhere super quickly, right?
So I think maybe to be a little bit more precise in my language, what I mean eventually by more local niches is more local chemical niches, because in this case, that's all that matters. So as long as the chemicals are, you have niches with different chemical properties and situations, then that would create more opportunities for coexistence.
Okay, thank you.
Yeah, thank you for a nice presentation. This was in 1D, right?
Yes.
Do you have any insights about what happens in higher dimensions? Because, well, nasal cavity is not entirely 1D. It's probably 2D.
Right. So there are, I guess the short answer is that we haven't done it systematically. So anything I say is a speculation. I think the general ideas will apply in 2D as well. Again, if you have more local niches, you have more opportunities for these. There are some differences because you have also more freedom in moving around in the 2D, especially in cases that we intentionally ignored any active movement or migration in our system. So incorporating those would be very important, especially in 2D, because there is more opportunities for dynamics outside of the simple cases that we've studied before here.
Other questions?
Yeah, thanks, Babak. I think I might have missed some of what you're saying. So when you say the trade-off between more diverse pole versus small poles, so the total diversity of these two scenarios are the same. Is that right? So either you have, say, one pole of 100 species or 10 poles of 10 species. Is that what do you mean?
So in this, let's say, where did it go? Sorry. In this simple case, we have 5 species.
Yeah.
And in this case, we've waited after we start from an initial condition that intentionally they are just segregated in space next to each other.
So 5 of
right.
5, right?
And after 30 generations, you see that there is like maybe 3 of them overlapping here. And you don't see 3 overlapping here because the dispersal is so slow. So here you end up with only overlap at the interfaces of different types. So you get maybe 4 niches here. You get maybe 2 niches here, essentially, because things are spreading so much. And then it becomes sort of enumeration. Because getting smaller communities is much more likely, these ones win over those ones. Because if you add 3 or 4 species together, it's very unlikely to still get 3 or 4 of them coexisting together. Does that make sense? So that probability in our model exponentially drops. So if you put 5 together, it's exponentially less likely to get 5 than 4 than 3 than 2. So if you get fewer of larger pools of microbes, you end up with smaller overall chance of coexistence compared to the case that you get many of these smaller pools of 2 species interacting with each other.
Hey, Babak, great talk. I was wondering, have you looked at like diffusion constants for all these things? And in particular, I imagine if I have diffusion, if let's say the diffusion constants for all of my like resources or my mediators are drawn from some distribution, if I have uneven diffusion constants for all of these, do you get actually more spatial niches because like the different gradients sort of interact in a weird way? Have you tried that?
We haven't tried it, but my speculation will be that the fastest one will, I guess, will spread faster, and then you end up sort of deciding maybe the intermediate levels would describe what you will end up seeing in that environment. We haven't looked at, so I should, I guess, be more careful about describing. We haven't tried picking diffusion constants that are different for different organisms or different mediators. We assume the same number for all of them here, but I think it would be interesting to look into how having something that disperses faster or having some mediator that diffuses faster would affect the community structure. We haven't done those investigations yet.
As an additional question, and I think you largely said this already, but my brain's just catching up. It seems like this is a nice contrast to what I think of as the central message of like invasion ecology, where you expect diverse communities to be hard to invade, and you're finding the opposite result. And I mean, for different reasons, I think, but would you agree with that statement?
So we have a paper out that talks about the sort of those trends.
Okay.
What we find is that for smaller communities, it's harder to it becomes harder as they get more rich. For really large communities, it becomes easier to invade them because there are members that are close to extinction anyway. So things are not monotonic.
Okay.
And I don't think we are contradicting anything necessarily, but there is a trend there that I think it's worth exploring even a little bit more with spatial communities, and we haven't gone that far. We have done it only with well-mixed communities.
Great. We are next speaker can set up. Any last questions?
Did you find anything that was like anti-correlated with disruption, where like something would die, or was that just like not something that was interesting to look at?
Things that are anti-correlated to disruption. So I guess maybe I won't exactly answer your question, but we see disruption in communities that are already sensitive to perturbation. So those communities, the introduction of invasion, instead, if you had changed the population structure a little bit, or you had changed something about the interaction coefficients, those species would have gone extinct. And in this case, our invader is the perturbation that causes that extinction. So I would imagine if you pick parameters that give you more stable communities, then you end up having things that don't get perturbed as easily.